In this video, I'd like to talk about how to form phospholipids. Phospholipids are very important structures as they're found in our cell membranes. So let's discuss how can we actually produce these phospholipids. The first thing that I want to mention is that this structure that I've drew right here is known as a phosphatidic acid or a phosphatidate. And this is a very, very important molecule because this will be used to form our phospholipids. If you recall, this molecule is shared with the production of triacylglycerides. We actually began with glycerol 3-phosphate and then added 2 fatty acids sequentially, right, using acyltransferase, and now we form this phosphatidic acid. This molecule will allow us to add a head group. Now, phosphatidic acid is actually a phospholipid. It's the simplest phospholipid. But to form other phospholipids, you need to add a head group. And that head group is often an alcohol, like an amine alcohol. But it can also be an amino acid, like serine, or a sugar, like inositol. So how do we add the head group so we can actually form different kinds of phospholipids? Well, there are 2 methods of doing that. Let's discuss the first method. In the first method, we actually begin with this molecule right here. This is known as DAG-CDP. If you recall, DAG is glycerol bound to 2 fatty acids. But now we have this extra group that's attached to it. This is known as CDP, or cytosine diphosphate, because it has 2 phosphate, a ribose sugar, and a cytosine base. So this is a CDP structure that's attached to our DAG, and this is important to activate the DAG. So now DAG is active and it can actually participate in the formation of a phospholipid. We have a polar head group that we want to add to this structure, and the polar head group has an OH. The oxygen is a nucleophile, and the phosphate on the CDP structure will be nucleophilically attacked by the oxygen of the polar head group alcohol. And when it will be attacked, we will form this structure right here. The O is going to attack this phosphate and it's going to release this portion here. The portion that's going to be released is actually CMP. CMP leaves and we are left with this structure, which is a glycerophospholipid. So this is the first method of forming our phospholipid. We start with this DAG, diacylglycerol, bound to a CDP so it can be activated, and then the polar head group, specifically the oxygen portion of it, is going to attack the phosphate of this molecule and is going to release a CMP and liberate our phospholipid, our glycerophospholipid. So this is the first method of forming phospholipids. Now let's look at the second way that we can form them. In the second method, right over here, we actually begin with a different molecule. We begin with a molecule that's known as DAG, diacylglycerol. And what's going to happen next is this right here, CDP head group. So here we have a CDP bound to the head group, is actually going to be involved in this process. So as I mentioned, we have our CDP alcohol head group and we have our DAG. So the oxygen of this DAG molecule is going to attack this phosphate of the CDP head group molecule and is going to liberate again a CMP. So a CMP is going to leave and again we're going to form our glycerophospholipid. So we can start with either a DAG bound to a CDP or to just a regular DAG molecule right over here. And this is really, really important to understand because there are 2 methods of forming it. So let's discuss what we have just learned. In this first method, we have DAG-CDP plus a polar head group forming our phosphoglyceride. And CMP will be lost in this process to form our phosphoglyceride. Now, another thing I wanted to mention is how do we get this CDP DAG in the first place? Well, we're actually going to start with a phosphatidic acid. So in order to create this molecule, we're actually going to begin with a phosphatidic acid. And we're going to add CTP to it, CTP. And in the process, we're gonna liberate our CDP DAG and a pyrophosphate. So we need this phosphatidic acid to form our DAG-CDP molecule in the first place. In the second method, we start with DAG and we add a CDP head group to it to form our phosphoglyceride. Again, a CMP is going to be lost to form our phosphoglyceride. Now, what's going to happen in this method, let me actually put this in a different color, is we're going to have a polar head group plus ATP, and we're going to need a kinase. We're actually going to add the phosphate to our polar head group. So we're going to have a polar head group now bound to a phosphate plus ADP. So notice how this process actually requires ATP. Once we have our polar head group, we can come right over here and we can add our CTP to it, and then we form the polar head group CDP and a pyrophosphate. The polar head group CDP is our substrate that we had right here that we're going to combine with DAG to form our glycerophospholipid. Notice how to form this molecule we need ATP. So ATP can be also required in this second method. But in both of these methods, we require a CTP molecule to form our starting product, whether it's the DAG-CDP or whether it's the CDP head group. We need a CTP molecule, a cytosine triphosphate or a cytidine triphosphate, in order to create our starting product that we're going to then react with something else to form our glycerophospholipid. So again, this is an important point to understand, because this phosphatidic acid is absolutely crucial, but it's used specifically for this first method of forming the glycerophospholipid. Anyways, I hope you found this helpful.